Welcome to this explainer. Okay, so we always hear DNA called the blueprint of life, right? But think about it. A blueprint literally can't build a house on its own. You need the whole shebang, an assembly line, specialized workers, and strict quality control. So today we're diving straight into exactly how that molecular factory operates inside your cells right this very second. We're talking about the mind-blowing biochemistry of protein synthesis.
It really is a wild question. I mean, how on earth do we get from a basic four-letter genetic alphabet of nucleotides to the incredibly complex proteins that make up, well, us? This whole biological mystery is solved by this insanely efficient, highly coordinated dance that's happening millions of times inside your body every single second. It's all about translation, taking one language and flawlessly converting it into another. To kick things off, let's look at the actual math behind this blueprint. We're dealing with a redundant triplet code. Basically, 64 unique codons dictate just 20 amino acids. Because proteins are built from those 20 specific amino acids, a 1-to-1 or even 2-to-2 nucleotide code just wouldn't cut it. So nature settled on a triplet code. 3 non-overlapping bases make up one codon, kind of like a 3-digit combination lock that yields exactly 64 possible combinations. All right, let's jump straight into section 1, deciphering the genetic code, and see exactly how this cellular blueprint is established and read. When we map out these 64 codons to their corresponding amino acids, the coolest part is how strict the reading rules are. It is read strictly in the 5' to 3' direction on the messenger RNA, and it's beautifully non-overlapping. You'll also notice the code is redundant or degenerate. So ACU, ACC, ACA, and ACG, they all code for the exact same amino acid, threonine. But hey, redundancy doesn't mean it's flawed. Each specific codon specifies only one amino acid. ACC will always mean threonine. It's never going to suddenly code for serine. And honestly, this code is so absolutely crucial to life that cracking it earns Nirenberg, Corona, and Hawley the ultimate historical prestige, the Nobel Prize. They literally deciphered this complex nucleotide dictionary and completely transformed modern biology. Corona actually synthesizing nucleotide polymers with defined sequences, that was the skeleton key that finally broke the whole code wide open. Just a huge, game-changing moment. But this blueprint actually introduces a bit of a mathematical mystery, which brings us to section 2, the wobble hypothesis. Okay, remember those 64 codons we just talked about? Well, 3 of them are stop codons. That leaves exactly 61 codons actively coding for amino acids. Now, we know transfer RNA, or tRNA, acts as the physical adapter reading these codons. So logic tells us there should be 61 different tRNAs, right? Well, no. The math actually doesn't add up. Most organisms only have between 30 to 35 tRNA genes. That's a massive discrepancy. It means nature basically had to bend its own rules to make this factory run. So here is the brilliant workaround. Nature isn't perfectly rigid. Francis Crick proposed what's known as the wobble hypothesis. It explains that while the first 2 bases of the codon form those super strong, standard Watson-Crick base pairs with the anticodon, the rules at the 3rd position are, well, they're a bit more relaxed. This is a genius evolutionary shortcut. It literally allows a single tRNA molecule to recognize and bind to more than one specific codon. This relaxed base pairing physically happens right at the wobble position. That's the 3rd position of the codon matching up with the 1st position of the anticodon on the tRNA. The tRNA is oriented 5' to 3', and the mRNA is antiparallel. Because of this spatial geometry right at this specific spot, standard hydrogen bonding rules get effectively bent. It allows for non-canonical base pairing. Pretty wild, right? And to pull off this flexibility, tRNAs sometimes use an uncommon base called inosine. Inosine forms weaker hydrogen bonds. So if it sits at that wobble position, it basically acts like a skeleton key, pairing loosely with uracil, cytosine, or adenine on the mRNA. This is exactly how a single tRNA anticodon can successfully read 3 totally distinct codons. All right, with the blueprint and the wobble sorted out, let's watch this factory power up in section 3, the assembly line stages. Just like any highly efficient assembly line, protein synthesis drives forward relentlessly through 5 coordinated stages. It kicks off with activation. That's where the tRNA gets charged with an amino acid. This sparks initiation, locking the mRNA and that charged tRNA onto the ribosome. Then, boom, the machinery fires into elongation, adding link after link to the chain until it hits a stop codon. That's termination, where the new chain is released. And finally, the raw protein goes through folding and processing to get its ultimate functional shape.
Let's move into Section 4, the major cellular players, and actually meet the specialized workers keeping this whole line moving.
We mentioned them briefly, but tRNAs really are the ultimate adapter molecules. Without these little delivery trucks bringing in the exact right amino acids, those mRNA instructions, totally useless code. They've got a specific anticodon on one end to read the mRNA, and they carry the corresponding amino acid right on the other end.
The tRNA actually has this incredible clover leaf architecture. Up top, you have the amino acid arm. Then you've got these specialized side arms, the D arm and the T-Cy-C arm. These act as structural scaffolding, ensuring the whole molecule folds perfectly in 3D space. And right at the bottom is that vital anticodon arm, with its wobble position perfectly exposed and ready to interact with the mRNA.
Next up, we have the heavy machinery itself, the ribosome. This is the literal factory floor where the magic happens, reading the transcript to build the protein from the N-terminus to the C-terminus. It's made of large and small subunits that clamp together around the mRNA, scanning along in the 5' to 3' direction until it hits that AUG start codon. And here's a fun fact. There are actually two distinct tRNAs for methionine. One is an initiator that binds to the empty ribosome at the P site to kick things off, and the other one only binds after synthesis is already rolling.
But look, a factory is only as good as its quality control, right? Which brings us to our final stop, Section 5, quality control and proofreading.
I want to introduce you to the unsung hero of translation, amino-acyl tRNA synthetase. This enzyme is required to catalyze the attachment of the correct amino acid to the 3' acceptor stem of its cognate tRNA. That's what it means to charge a tRNA, using ATP hydrolysis for energy. But its job is way more intense than just activating the amino acid. Here is the absolute craziest part. The massive ribosome, it is completely blind to mistakes. It's an amazing linking machine for sure, but it blindly trusts that the tRNA adapter brought the correct part. It does not check the amino acid. It relies 100% on the syntheses. The syntheses has this incredible double sieve editing function. First sieve, structurally related amino acids are charged, but if a mistake sneaks through, say valine accidentally attaches to a threonine tRNA, a second sieve editing site catches it and physically hydrolyzes the mistake. Proofreading is its absolute most critical job. It is just wild to think about. Every single physical structure, every enzyme, every cellular process making you who you are right now relies on this giant blind ribosome factory assembling pieces at breakneck speed. Our entire existence hinges on this microscopic quality control inspector, the amino-acyl tRNA synthetase, just tirelessly catching errors so the whole operation runs flawlessly. Without it, the translation of life would literally be genetic gibberish. Definitely something to chew on the next time you marvel at the complexity of the human body. Anyway, thanks for hanging out with me for this explainer, and keep staying curious.